Okay, so now we're going to begin thinking about foundations of molecular design, and in particular, we're going to be thinking about construction. So, in molecular construction, this is really how do we build molecules on a computer? And there are some key areas that we're going to cover. So, we're going to first cover atom-based, so this is building molecules atom by atom. Then we're going to cover fragment-based, and then we're going to cover reaction-based. And this, hopefully at the end of this short lecture, you'll be able to understand the differences therein. So to begin with, we think about molecular construction. We can really think about molecular construction operating on a spectrum, and that spectrum we look to balance control and synthesizability. So on the left-hand side here, we have atom-based, and in atom-based, we have infinite control, so we can position an atom exactly where we want on the molecule. However, the issue is that we may not be able to synthesize that molecule successfully. And that is really where we have to start to think that there is a balance between being able to reach anywhere in the chemical space versus actually being able to make that molecule in practice. So in the middle then we have fragment-based, and fragment-based tries to couple large building blocks together, and then from that we get a little bit less control, but we get more synthesizability because we presume that in the middle our fragments, our building blocks, should be things that we can actually make or we have to hand in the lab. And then finally, at the far end here, we have reaction-based, which has a maximum amount of synthesizability. However, one of the trade-offs here is that in the reaction-based example, we have far less control. So we can't dictate exactly where we want to place, for example, a halogen in a strained ring system because we simply cannot synthesize it. We do not have transformations that will be able to construct that molecule. So we have to think about it as a spectrum where control is minimal for reaction-based but maximum for atom-based, and then on the other hand we have synthesizability, which is minimal for atom-based but maximum for reaction-based. So to jump in then into atom-based construction really, as I mentioned previously, gives you maximum control. And how does it achieve this? Well, in atom-based design, what we're really thinking about is how do we build a molecule atom by atom, bond by bond. And this means that we have all of the periodic table to play with. So this is a nice sort of heat map over the periodic table. And at the top here is the compounds that are, or the atoms that are present in, I believe, all of the substances in PubChem. These are all the chemical substances. And then beneath that then is all the atom types that belong in molecules. And you can certainly see that for the majority we are in this region of the periodic table: carbon, nitrogen, oxygen, fluorine, the halogens, silicon, phosphorus, sulfur, chlorine, etc. And on the other hand, we also have to now start to think about, with atom-based construction, we have to start thinking about bond typing. And so we add an atom, but we also then have to carefully think about its corresponding connectivities. So now the connectivity we're starting to worry about, we're starting to say, okay, given an atom and its bond, how many different types of bonds can we form? So for example, we can put a carbon atom here, but does that carbon atom, is it part of a ring? Is it part of a chain? Does it have a single bond, a triple bond, a double bond? Is it aromatic? Is it undefined stereochemistry? All sorts of different considerations. So certainly atom-based construction allows us to have an awful lot of control. Now, with atom-based construction, there then forms three sort of sub-operations that one can do. And this is really now thinking about how do we actually start to apply these transformations? How can we add an atom or add a bond to a molecule? And so we can start here, for example, from bromobenzene, and we transform it into our chemical graph as described in the previous sort of video. And what we can see is that we can add an atom, and adding an atom to the chemical graph, we now get dibromobenzene. We could delete an atom, and that deletion of an atom would return benzene on its own. And then finally, we have substitution. This is where we can switch an atom in. So we could have, for example, bromine goes to chlorine. And this would give us chlorobenzene. So at each step, when we're thinking about atom construction, what we're saying is how can we modify our small molecule? And we do that by usually transforming into a graph and then modifying the graph directly. And at each step, we have to then worry about things such as valence. And that is because we might add a carbon, but if we just add a carbon, of course, carbon often comes with a lot of other bonds. So, for example, we have to then worry about what is the carbon attached to. So an example of this is that, for example, we can take benzene, and if we wanted to add a single carbon to that benzene, well, if we just added it directly, in this case, I have included all the explicit hydrogens, we can see that the addition of the methyl, if we just added it directly, well, one problem that we would have is that this carbon would now have more than 4 bonds. It would have 5 bonds, 1, 2, 3, 4, 5. And what that would mean is that now it has an incorrect valence. So naturally, we would have to delete a hydrogen to be able to then form methylbenzene here. Now, the key thing is that under almost all circumstances, we are almost always having to do chains of operations to be able to get the right valence, because adding an atom or deleting an atom may lead to issues associated with propagation of each transformation. Okay, so next up, then we're starting to think about fragment-based methods. So in fragment-based methods, this is very much where we are seeing that the fragments are individual building blocks, and these individual building blocks are often taken from pre-existing molecules. So here, for example, in this example, we have a molecule, and that molecule we then go ahead and fragment. And this fragmentation builds up several building blocks. And these building blocks can come from various different classes. For example, we can have ring classes where we're capturing all the rings that were present. We can have linkers. These are usually alkyl chains or functionalized chains which link together to individual components. And then finally, we have sort of side chains, and these are often seen as decorators. So these are where do we decorate our molecule and how do we sort of achieve this. And the interesting thing about fragment-based approaches is that it's pretty hard up front to know what constitutes a useful fragment. So, for example, it might be that, for example, fragments with multiple substitution points are actually quite hard to use because we have to then bind on multiple components. We may find that linkers may not actually be synthesizable. So we have to start to think very carefully about what constitutes a good fragment library, what constitutes a correct fragmentation approach. So assuming that we can do all of this, we should be able to take a collection of molecules and transform them then into a fragment library which we can then use. Now, I want to just say briefly that in vendor catalogs, you may come across fragment libraries, and fragment libraries are often highly functionalized small molecules which have particular exit vectors which can be modified. And usually these fragment libraries are used as part of a screening campaign to identify good starting points for drug discovery. In this particular example, these are fragments in terms of molecular fragments. This is really where we're starting to think about, okay, their individual attachment points. Again, just to kind of cover the sort of nomenclature between the two different approaches. Okay, so now we're going to go on to fragment-based construction methods, and how can we start to then clip together these individual fragmentation-based approaches. And the key thing to keep in mind here with fragment-based construction is really starting to think about how do we join them together and what are the different macro strategies. So the first one is growing, and fragment growing is the most obvious strategy. This is where we decide, okay, somewhere on the molecule, this is going to be where we're going to grow off. We're going to, okay, identify an atom, and we're going to try to modify that and grow out in 3D space. And this is called fragment growing. Now, fragment growing is often an attractive strategy because often there is a certain region of a pocket or a particular interaction that we want to try and gain, and by doing that, this is a natural sort of extension. We're trying to extend the molecule to be able to reach that space or reach that part of the pocket. On the other hand, the next sort of approach is linking. In linking, we take two fragments, sometimes positioned in the binding site, and what we try to do then is we try to link these together by using a linker as described previously. So what the idea is that we have fragment A, fragment B, and then we have some kind of linker that exists between the two. And the linker that exists between the two, that can come from many different sources. However, the key difficulty often is that this linker will have several rotatable bonds. Okay? And what that means is that those several rotatable bonds will often lead to these individual fragments having a free flexibility. It means that they can rotate as and how they wish. And that means it can be very difficult to be able to keep the same geometry of fragment A and fragment B in a binding site. Now, this is a 2D representation, so it's hard to describe, but you can think that in this example here, this fragment in green can rotate as it wishes, and this doesn't mean that, just because we have two fragments, both of which have shown binding modes, it doesn't mean that when we have this linker between the two, that will be maintained. The final method is a little bit more uncommon, and that's called fragment merging. And in fragment merging, what we do is that we take two fragments and we try to identify a substructure which is common between the two. Here, for example, both of them contain this 5-membered ring. And we try to then overlap them, and then we build a sort of merge structure as shown here. And we can see that that red sort of middle fragment was common between both of them. And this is an attractive strategy for several reasons. The first reason is that it usually gains the benefit of both fragment A and fragment B. It is a low molecular weight, so that means we're not growing out of the molecule. And what we are doing is we're trying to take the sort of best of A and the best of B and then fuse them together. Now, that will often lead to a molecule which has a high degree of ligand efficiency. However, there is a trade-off, and that trade-off is that this molecule can still rotate. So we can still have some degree of rotation. If this is not, for example, a double bond, we would then have free rotation of this ring system, and that again means that we can end up in a situation where just because fragment A binds well and fragment B binds well, there's no guarantee that the fragment on the right-hand side will also bind well. So, as I highlighted, fragment-based construction is really about clipping different fragments together and being able to then build novel molecules. However, one of the key issues with this is still synthetic tractability. And I've kind of shown an example here. So here's the collection of fragments that are shown on the left-hand side. And I went away and clipped them together. And we can see that sometimes we can build molecules that are very synthesizable. So this is a molecule that absolutely we would believe should be synthesizable in the lab. But on the other hand, we can make molecules that are certainly not synthesizable. These are molecules which are quite complex. We can see that this is a macrocyclic structure here. And this macrocycle is going to be a challenge for a synthetic chemist to make. So even though our individual building blocks are indeed synthesizable, tractable, it doesn't necessarily mean that the final endpoint will be. And this is again the comment, which is we are sitting on a spectrum between synthesizable and control. We get less control, but we get more synthesizability, but that synthesizability is not a guarantee that we can actually make the molecule in practice. Moving on then to reaction-based approaches. Reaction-based approaches is one of my favorite approaches to generative design, as reaction-based approaches rely upon synthetic transformations that actually chemists do in a lab. So at the top here, when chemists, for example, make molecules, they go away, run the experiment, and they will store the reaction information either in an ELN, so an electronic lab notebook, or sometimes in a paper lab notebook. And the idea here is that these are real transformations that chemists have done. And what we can do is we can say, okay, well, what reactions, what transformations are chemists applying, and how can we encode those transformations into virtual reactions, which we can apply, say, using a Smerk's transformation or the condensed reaction graph, as shown previously in the section on cheminformatics. And what this allows us to do is that, for example, if we have an amide type reaction here, what we can do is that we can then begin to clip together large building blocks and actually then synthesize the molecules using the same transformations that chemists would do in practice. And this is really attractive because we know that chemists can do, for example, here, an amide coupling. So it should be possible that so long as we have the individual building blocks, the left-hand side and the right-hand side, in this case of the molecule, we should be able to perform that reaction. And by doing so, this means that we get a very high propensity to be able to have molecules which are synthesizable. But again, there is not a guarantee, because sometimes reactions require long-range functionality, or there are issues associated with cross-functionalization, which makes things difficult. Right, so reactions themselves, and in particular reaction templates, which we use for reaction-based molecule construction, are sourced from reaction examples. So here in this example, I will show multiple amide couplings, and what we get is, if we generalize this sort of reaction transformation, we get this Smerk's transformation here, which we can then prospectively apply to new acids and new amines to be able to form new products. And this is the attractive part of reaction-based generative design, because what it allows us to do is it allows us to be able to build in silico new molecules using reaction transformations that in the lab chemists will be able to make. So with reaction-based construction, then the extension of this starts to become a loop. So we can encode collections of reaction transformations as Smerks. We can then use them to design new molecules. We can then pass those new molecules with a synthetic scheme to the chemist, which will then make the molecules if they can. They will then store the reactions in the ELN, and then we can go ahead and we can extract those reactions again and form a reaction database, which can then be used to design yet more molecules. And this then provides a sort of closed loop where chemists are making molecules, and then the designer is taking those templates and trying to then generalize them to be able to build yet more molecules, and thus the loop continues. And this makes reaction-based construction very nice, because it can actually directly connect with lab synthesis. Finally, I'm going to wrap up this video with a brief description that each construction approach really does have its distinct advantages and limitations, whether that be in the case of control or synthesizability, but also in terms of the computational complexity of each of the methods. Moreover, when we are thinking about where should we use each approach, well, that very much depends where in the project we are. Sometimes we want to be able to explore completely a particular space, and other times we want to be able to have a much more narrow set of molecules which can then be forming a library. So it very much depends upon where we are in our project, what is the information we have available, and then what is the kind of budget in terms of how many molecules can we have made, what are we trying to look for in terms of compounds that are designed, and this then will ultimately dictate what construction method we use and why.